All right, here's where we're going to find just kind of hints and tips on how to make a good lab report for IPESS when we're talking about our productivity lab report. You're going to be graded on kind of bits and pieces of the IA rubric. The first part we're going to look at is the data collection portion. You're going to be graded on aspect 1 and aspect 2 of the data collection. The reasoning for that is we really didn't test an independent and dependent variable. What we're doing right now is taking a look at what kind of procedure could we use to collect productivity and getting a feel for it. Is it good, is it bad? So we're going to look at aspect 1 and aspect 2 of data collection. So let's take a look at aspect 1. This is your data table. So while we've been doing this lab, and I realize it's been over 2 weeks, you need to collect certain data, take a look at what's going on with your lab. So of course there's the quantitative value. This should be in some sort of data table. And on your data table, you have to remember you need a descriptive title. Saying productivity lab, I'm telling you right now, is not a good title. You need to include your uncertainties for your measurements. So if you measure the height of the grass, you want to look at the uncertainty of your ruler. If you measure the mass of the grass, you want to look at the uncertainty of the scale. And the big thing, and I want you guys to remember this, when you look at your rubric, look at the checklist, it asks for qualitative data. So you could have drawn a picture. You could have taken a picture of the grass before and after. You can describe what the grass looked like between your two different sets. When you had the light versus the dark grass after a week, did anything change between the two of them? And this qualitative data, it's great that you write it down, but you need to make sure when we're talking about it later, you use this data to describe any anomalies. So if I looked and I was like, man, my numbers are really off, it didn't work, I'm going to go back and look at my qualitative data to see, well, what would help me explain this? What would help make it make sense? All right, so that's kind of what are you collecting, why are you collecting it? The next part is looking at aspect 2, which is what are you going to do with these numbers that you have collected? What is their value? Do I need to do anything to them? You guys need to calculate and show your calculations. Do not just kind of write down what the numbers are. You need to calculate NPP, which is your net primary productivity, your GPP, and you need to calculate your respiration. Sorry for my handwriting, guys. And some folks ask for a refresher on how to calculate them, and I will happily give that to you. So NPP is all the photosynthesis that's done minus respiration. And I'll do this in a different color. So let's take a look at NPP. It's going to be your light final value minus the initial grass value we had. And don't forget your units on that, and we'll talk about that in a second. Respiration is all the respiration it did, and hypothetically, and I say this hypothetically because I don't think it works for the majority of our groups, is that the only thing this plant is doing is respiration. And we want it to be a positive number because that's how much respiration it was doing. So you're going to take your initial value of grass, and of course we're using only dry here, only the dry values, minus your dark final. And the next part only really works is when your initial value is the same for both light and dark, and for us it was. But I want to go through and show you how you would do it without having the initial value is the same. So let's go ahead and take a look at GPP. Gross primary productivity is NPP plus your respiration. And we can just take these equations up here and fill them in. So if NPP is light final minus initial, and I have to add back respiration, which from up here is initial minus dark final, That's going to give us, if we do some math, and I'm just going to do a little bit of algebra here. I have a negative I value, I have a positive I value, so those actually cancel out. So when I'm doing this, I end up only having to use the two numbers, which is my light final minus my dark final. But say I was doing dissolved oxygen and I had two different values, I couldn't do this easy light minus dark. I really have to go back and use this equation up here because I can't make that assumption anymore. But for us with biomass, since it's a lot easier and we had to take it out and kill it, we had one initial value for our grass. The last thing I want to talk about is units. Units were kind of our sticking point when we were doing our quiz. A lot of folks have been getting better, but you have to think about there's data we didn't collect because I purposely wrote a terrible procedure. But there's also data you needed to keep in mind, or things you needed to keep in mind for having units for this. Things like we spent 8 days letting the grass grow or not grow, so it was over an 8-day period. We calculated everything in grams. And the thing we didn't actually take a look at is the area it was in. Everyone kind of trusted me to do it in an egg carton. Well, what is the area per each section of our egg carton? And this right here was kind of a sticking point. And I was wondering if anyone's going to pick up on it. And if you didn't, that's perfectly fine. Just don't include that in your units. Or if you do, you make a third of an egg carton your unit, which is not really scientific, but it's going to have to be the best way it can be. So you've done your calculations, you've kind of done all that you can do for the data collection and processing. Because we weren't testing an independent and a dependent variable, you're not really making a statement. So when we take a look at the next part, and you can't really make a graph, which is conclusion writing, and where you're looking at your errors and limitations, you can make a conclusion. It's not like we did the effect of fertilizer on productivity, the effect of temperature on productivity. That's not really what we had time to do, but it's definitely something you could consider for an IA at the end of this unit. So you're not making a conclusion, so that means we're not doing aspect 1 of the rubric. But we are going to do aspect 2 of conclusion, where you really are looking at, well, let's take a look at my procedure. And I won't take it personally, I wrote a bad procedure. So the things you want to look at is, the very first thing you want to do is uncertainties. If I had plus or minus 0.01 as an uncertainty, and the difference between my light and dark was plus or minus 0.01 or plus or minus 0.02, well, then I didn't have any difference at all. So you want to look at your measurement uncertainties first. The next thing you want to look at is kind of those data points that didn't make sense. And you want to think about, well, why? Why was dark heavier than light? Why was the dark value heavier than my initial value when it should have been losing mass the whole time? And to do that, you can look at human error, of course, but I don't want to see, oh, I mixed them up, or, oh, I forgot to press 0 on the scale, because it's not necessarily the only reason. The procedure was not written perfectly, and we didn't kind of go through the procedure quickly. So I want you to evaluate the procedure as to why. So things like, I told you to plant grass, but I didn't tell you how many seeds. So we could have had a different amount of grass in each section. I told you to put foil on it, but I didn't tell you how. So some you have crazy grass, and grass naturally will grow towards the light. So you want to look at the procedures, you want to look at your errors, and of course you want to look at things like limitations. So limitations are things you could not control based on the resources you had. So maybe we should have let the grass grow less the first 2 weeks, just do 1 week of growing. We should have let it do in the light dark for 2 weeks. You can talk about the egg carton. A lot of folks had the roots grow into the egg carton because it was starting to break it down. So you want to look at all these kinds of things. What happened? What was wrong with the procedure? And you want to say what's wrong with it. And once you're done with evaluating the procedure, for each thing you kind of mentioned, you want to look at its possible effect on the data. So if I just say that the roots grew into the egg carton and that messed up my data, well, that's not good enough. How did it mess up your data? Did it mean that things were heavier? Did it mean that things were lighter? What was lighter? My initial value? Did it make a difference between my initial and 2 weeks later or 1 week later? What happened with my light dark values? And I want you to tell me, was it significant? Was it enough to really mess it up? Okay? So that's all about aspect 2. And unless you're making this connection right here, what was the effect on your data? How did it mess up your experiment? You're not really getting full points. You'll be lucky if you get a 1 out of 3 on that, or 1 out of 2 on that section, because you're not taking that moment to think, well, was it really important that that happened, and why was it important that that happened? The last thing we want to do is aspect 3, which ties into aspect 2 very easily, which is when we're talking about, well, let's talk about next time. So you have a list of mistakes, right? You just made it in the previous paragraph. The next thing you need to do with that list of mistakes is, how would you fix them? What could I do? What other resources could I use? Should I have fixed the time I used? Should I have been more careful with the tinfoil? Did I water them every week? How do I fix all these mistakes that happened? And of course, you have to connect it back. Well, will that solution make a difference in my data at the end? So will this fix help your data, or how will it affect your data? So if I decided to, instead of using an egg carton, I use a plastic container to grow my seeds that had drainage at the bottom, well, maybe my roots wouldn't have grown in. It would have been easier to pull out, and I would have higher values all around, and maybe in the first 2 weeks, my roots didn't go into the container, but the last week it did, so that might help my values change that way. That might help, my NPP and GPP fix itself. And the last thing you want to do is, when you're fixing them, are my solutions realistic? You don't want to do something where I'm going to go spend a year in the Amazon looking at different kinds of grass and kind of doing all kinds of crazy stuff, but maybe I could have gone outside. We could have grown grass outside, but then you have other factors. So if it's not realistic, if it doesn't kind of fit in those limitations, and you want to take a look, what makes it realistic? Well, the value of what you want to do. Don't be trying to spend $1 million, and the time it takes. You don't want to be spending, 3 months on an experiment when you could do it in 1. So that's kind of the things I want you to look at when we're writing our lab reports this time. And hopefully this kind of puts ourselves, instead of the 0 or 1 range, it puts us into the 2 range for each section. And I'll try and get these back. But I want you to think about, if you had to do your own experiment about productivity or about plants, use this as a way to make a better procedure. So think of this as, how will I make a better procedure, and take this as an opportunity to see, well, do my ideas make sense, and can I follow through with them later? So think of yourself actually doing these fixed procedures that you have made, and that'll make your realistic section, can I give you, is it realistic, a lot easier, because you actually would have to do it. So please